Hi, my name is Kareena Villalobos, and for my 918 final presentation, I will be discussing the role of BMP4 at multiple development times. BMP4, or bone morphogenetic protein 4, has been studied by scientists since the 1960s when it was initially discovered for inducing ectopic bone formation, hence the name. Eventually, in the 1980s, researchers were finally able to study BMP4 biochemically after they were able to characterize and clone it. Since then, BMP4 studies have involved inducing mesenchymal stem cells to differentiate into a variety of organ cell types that include bone, lung, muscle, and ureteric cells. Recent studies have shown we can use BMP4 to create therapeutic regenerative solutions for medical issues. A few examples include treatment for IPF, or idiopathic pulmonary fibrosis, which causes scarring of the lungs. Another example is how BMP4 is connected to zinc during immune cell recovery, particularly in the bone marrow. But in order for us as scientists and engineers to arrive at medical solutions, we first need to understand the fundamentals of BMP4, and namely how it is involved in different stages of development. So first and foremost, we must know, what is BMP4? Well, BMP4 is a secreted signaling protein encoded by the BMP4 gene that is found on chromosome 14, which is boxed in red. In particular, it is found on the Q22 to Q23 section of the chromosome. It belongs to the transcription growth factor beta superfamily, or TGF-beta. So here we can see many BMPs in a family tree shown by their closest to each other. Many members of this superfamily are known to play an important role in many developmental stages, namely embryonic axis formation. However, this presentation will primarily cover BMP4's specific roles in both early embryonic development through epidermal induction and embryonic dorsal ventral axis formation, and in later development through adult neurogenesis. In early development, when the blastula only has an animal and vegetal pole, BMP4 limits the extent to which neural differentiation in xenopus embryos occurs by inducing epidermis. This knowledge was discovered by isolating the animal cap of the blastula and seeing that when there is no BMP4 and the cells are separated from each other, neural tissue develops. On the other hand, when we add BMP4, it induces epidermal tissue. However, after further investigation, scientists realized that during early gastrulation, BMP4 actually induced epidermis by an inhibition process. So for neural differentiation to occur, the BMP4 was inhibited by noggin, cordon, and validation factors. But when epidermal formation needs to occur, BMP4 is no longer inhibited by the three factors. However, BMP4's role in early development after epidermal induction does not end there, as it continues to be expressed in ectoderm and mesoderm tissue. In later parts of gastrulation, BMP4 specifically aids in ventralizing mesoderm, which guides the dorsal ventral axis formation of the embryo. The first step in this process involves the induction of the roof plate and floor plate, which then start to express BMP4 and sonic hedgehog morphogens. Then, BMP4 and sonic hedgehog begin to act as inductive signals to each other, which means the two morphogens influence the developmental phase of the other. Once BMP4 is secreted from the roof plate and sonic hedgehog is released from the ventral portion of the notochord, the mesoderm is finally able to develop ventral face. This event establishes a dorsal ventral axis for the differentiation of later structures. After the early stages of embryonic development, BMP4 is involved in many organ systems as a secreted factor. However, I would like to leap into the adult stage of development to discuss why BMP4 is important for neurogenesis, which is the generation of new neurons that occurs throughout life, but only in the dentate gyrus of the hippocampus and the subventricular zone, adjacent to the lateral ventricles. Particularly, BMP4 and its antagonist noggin, which also played a role in epidermal induction, contribute to the modulation of neurogenesis in the adult hippocampus, thereby affecting hippocampal function. This occurs through the SMAD signaling pathway that transduces the necessary signals. First, BMP4 binds to two types of serine threonine kinase receptors, known as BMPR1 and BMPR2. Then, the BMP mediates signals via SMAD4, which is a protein. The signal continues to the SMAD complex, which then enters the nucleus to affect the transcription of target genes. This is what initiates neurogenesis from adult neurostem cells. BMP4 and its involvement in neurogenesis has been linked to being a potential therapy for sufferers of Alzheimer's disease. This is yet another study that demonstrates by understanding BMP4's role in early embryonic and postnatal development, we can further the biological and medical fields to develop applications that will push the boundaries of human life. Thanks for watching.